am AVS Priya, Department of Biotechnology. Today my topic is bioinformatics. Next. Introduction. Nucleic acid sequence databases, GenBank, EMBL, DDBJ. Next. Biological databases. Biological databases, libraries of life science information collected from scientific experiments, published literature and highly throughput experiment. Technology of computational analysis of bioinformatics tool. Next. Bioinformatics including genomics, proteomics and metabolomics and microarray, gene expression and phylogenetics. Biological databases, design, development and long-term management is the core of bioinformatics. Then simple databases using the biological databases and set of DNA and RNA informations. Then structural records and easily gene sequence analysis of bioinformatics tool. Next. Genome. First one of the genome and transcriptome and proteome. Genome is the complete set of DNA in the organism, including all genes of contains the information and structure, function, growth and reproductions of the organism. Then nucleotide sequence, adenine, thymine, cytosine and guanine. Then coding regions are non-coding regions. Coding regions to genes regulations and gene sequence analysis of bioinformatic tool. Then non-coding regions, regulatory sequence and introns. Then applications, overall human genomes and collected from the biological tool using the sequence analyzation and human genome project and phylogenetic tree analysis of the sequence informations. Transcriptome. The transcriptome is the totally complete set of RNA molecules. Then mRNA sequence, RNA sequence, totally analysis of the bioinformatic tool. Then cellular, tissue and particular species and tissue and contain to the overall collected the nucleotide sequence, adenine, thymine, cytosine and guanine. There are structure productions of the transcriptome. Then actively expressed and cell development stages and environmental conditions. Then RNA sequencing and microarray. First on applications, understanding the gene expression sequences and identifying the disease and biomarkers. So next. Types of biological databases. First one, there are three types of biological databases. First one, primary databases and secondary databases and derived databases. First one, primary databases. They are two types of nucleotide sequence databases and primary sequence databases. First one, GenBank, TDBJ and EMBL. Protein sequence databases, Swissport, TDBJ and PAR and GenPept. Then secondary databases. Secondary databases, they are two types. First one, protein structure databases and domain and modif databases. First one, protein databases, PDP, EDP and MMDP. Then domain databases, blocks and COG. Derived databases. Derived databases is the four types. First one, specialized databases and structural databases. Then metabolic pathway databases, gene expression databases. Overall nucleotide sequence databases, NCBI. NCBI is the best tool of bioinformatics. First one, BLAST, FASTA and using the genomic databases. First one, constants is the 3 centers of nucleotide sequence analysis in the size and available subdivisions. First one, download and then GenBank and then currently 17 divisions using the biological information. Then this databases overall collected from the GenBank, DDBJ and BLASTA and FASTA. Next, primary databases. Primary databases is the raw sequenced databases. First one, GenBank. Second one, EMBL and DDBJ. Website NCBI using the www.ncbi.nlm.nih.gov/omics. Then EMBL, European Molecular Biological Laboratory using the European biological technology. Then bioinformatic tool using the mainly NCBI tool. Then GenBank using the coding region and sequence analysis using the DNA and RNA sequence analysis. Then exchange data, daily process the synchronized. Then sequence analyzation and continuously proper alignment using the bioinformatic tool. Next. Primary databases is the experimental and raw biological data submitted directly and researchers using the usually gene sequence and identified and match sequence using the best tool. Then unprocessed and minimally processed data, then submitted by experimental analysis, then unique identifies and gene sequence identifications. Next. Secondary databases derived databases provide the process and annotated data, and primary source is the reference sequence using the transcripts, genes and proteins. Protein genes using the mainly unigene, best tool to the using NCBI. Then one transcript sequence, unique sequence, then unigene using the EMBL and DDBJ. Then genome annotation and visualization to the range of species. Next. Secondary databases mostly using to processed and interpreted and annotated information. First one include functional, structure and evolutionary annotation. Then integrate data using the multiple primary source, then analysis, comparisons and hypothesis generation. Next. Specialized databases. Secondary databases using the process, annotated data, then primary sources of the reference sequence. First one, genes, transcriptase and unigene. Unigene usually to the EMBL, TDBJ and structural visualization analysis of the secondary databases. Next. Tools mainly used to do the PDP, protein data bank, usually the structure predictions and rules and software using the PDP bioinformatic tool. Then sequence are arrangement and visualization and bioinformatics tool using the PDP usage and source. Structural databases visualization are 3D structural databases. First one, DNA and RNA. They help in understanding the structure, function and relationship of biomolecules. Then drug design and protein engineering and comparative modeling. Basic tool, basic local alignment, BASTA and FASTA using the use of the fast sequence alignment, and then analyzation equally participated in the bioinformatic tool. The databases provides the classification of protein, then classification of protein to arrange the 3D structure view and the evolutionary relationship. Next. CAT, primary databases, protein and DNA and RNA 3D structure, primary, large macromolecules and cryo and maps. Then primary, DNA and RNA nucleic acid structure, secondary, protein classification, fold and family analysis, and secondary databases, protein domains and structural comparisons. Next. Structural databases scope, structural classification of proteins using the secondary structural databases, organize the proteins and known 3D structure and classification of their evolutionary and structural relationship. Then understanding protein and function and evaluation. It is the manually correction and highly reliable. Then scope is the best method of using the BLAST. Next. Scope using the level and class. Class usually based on secondary structure and example mainly alpha, beta and fold. Then bold using the proteins with the major structure similarity using the secondary structure. Then superfamily, proteins and portable common evolutionary origin of replications and protein domain. Then PDP using the protein structure and protein source of the DNA structure and RNA structure, overall visualization tool the PDP using. Next. Database searching. First one, best tool using the NCBI enter to the GenBank using the basic tool of the BLAST and FASTA, and EMBL and DDBJ. Overall DNA sequence arrangement and similarity and mismatch. Any sequence arrangement and analysis of the using the DDBJ. DDBJ overall collected from the source of DNA data bank. Then BLAST basic local alignment tool. First one similarity identification and then sequence arrangement and then sequence rearrangement. Any other mismatch usually identify the scope method and BLAST. Next. Fast is the alignment process, order sequence alignment, then similarity sequence identified and database overall collected from the algorithm method, then different method using faster works is the short words and using the alignment and promising regions. Faster output percentage identify, e-value and alignment and database pairwise alignment and arrangement. Next, GenBank. GenBank is the most sequence searches. Then GenBank is the best tool using the bioinformatic tool. It offers the all exchange informations and major sequence databases analysis. Then widely used the detailed and help to the information collect already available into the GenBank. Then GenBank can be also used the during peak hours and identifications of sequence arrangement, then amounts of output and then completed more sequence arrangement. Then NCBI tool using the GenBank and collected from the DNA sequence and around the international val level using the NCBI level. NCBI center using the bioinformatic tool easily identified and sequence arrangements easily analyzations process main sequence and another sequence same or mismatch or matched sequence, any query sequence equally collected to the bioinformatic tool. Next, there are GenBank is the many tool available. First one BLAST and FASTA and database EST. BLAST basic local alignment and then database EST expressed sequence tags and ST sequence tagged sites. Submitting the sequence, GenBank is the very easy to required. Most journals of articles sequence are published. First one journals is the readers. First one researchers and using the best tool use the sequence alignment. Next, EMBL. EMBL is the good tool. First one using the limited amount of data and then easily trying to identify the sequence analyzation. Then EMBL is the mirror sites of located in the Europe. Then your connections will be more than equal information submitted to EMBL daily in the both GenBank and DDBJ. All of searching website using the humans and informations collected to the EMBL. Then EMBL molecular biology laboratory. It's the flat file. It's the many of multiple of various search engines. Next, DDBJ. DDBJ is the GenBank and EMBL is the beginning tools using the researchers and want to the databases and searching the interface. The DDBJ offers all its page is the Japanese as well as if you are more comfortable tool reading the Japanese version in the page, then very useful of DDBJ and DNA data bank overall collected from the DNA source. Then GenBank and EMBL both are collected the DNA sequence and arrangements. Next, overall collected from the biological tool. Best tool in the bioinformatics. First one, biological databases, important tool. Then understanding the easily and explain the GenBank, NCBI, DDBJ. Then primary database, secondary database and derived databases visually to the sequence identifications, then structural identification, then any other mismatch and matched sequence and comparative sequence analysis use the biological information, easily identifications, advanced development of bioinformatics tool. though any other newly identified sequence identifications and collected to the easily mismatch and any match sequence, first one identification, then any other process using the medicinal field, any other drug productions using the best tool and sequence analyzation to identifications and easily any disease caused the organisms. First one identification process, then researcher process, then sequence identified and mismatch sequence, any other query sequence equally to the stable of bioinformatic tool. Any disease presence in the bioinformatic tool using the easily cure sections of bioinformatic tool. Thank you.